Excellent. Yeah, like Nicholas said, I heard about this project 3 years ago, and I was, quite skeptical at the time that this is something that could work and that it makes sense to a clinical trial here. I've changed my mind considerably since then, and I think it's a very exciting experiment that I think is going to be probably the progenitor of a lot of such experiments to come, as it sounds like a lot of you folks are working on. But very excited to talk to you about PopVax. So PopVax really starts from this statistic here, which is that vaccines, despite recent controversies, have saved like a truly staggering number of lives. They're a very, very efficient public health intervention. Certainly there are adverse effects, there are issues, there are problems that individual vaccines might have. But in the aggregate, the number of lives saved, and this is really just childhood vaccines, is unmatched nearly by any intervention, in the last 30 or 40 years. And even during COVID-19, the vaccines, it seems like, saved a lot of people. They weren't perfect. They lost their efficacy over time, which is something we care about at PopVax. But it seems like a lot of people's lives were saved that otherwise would have been lost if the vaccines weren't deployed at the speed that they were. But unfortunately, vaccine development, despite being such an efficient way to save lives, is slowing down. Malaria vaccines are recently licensed over the last 5 or 6 years. You've seen RTSS and R21 Matrix M come out. These vaccines took 35 years from initial conception to licensure. And this is, as opposed to the 5-year timeline that the kind of previous generation of widely distributed vaccines took, in some cases at the height of Maurice Hilleman, for those who are familiar with his career at Merck. And these programs now, as a number of people have talked about over the last 2 days, cost an absurd amount of money. I think Lonis was mentioning this in his talk yesterday, but it often takes $1 billion+ to develop a single vaccine from conception out to an approval in a country like the US. And because these vaccines are dosed only a few times in a lifetime, they're actually very disfavored by pharmaceutical economics. Like it makes a lot more sense for a pharma company to make a drug that they can keep charging you for over and over again, like a GLP-1, than it does for them to make a vaccine that is preventative of substantial future chronic conditions. I don't think there are people thinking like this explicitly necessarily at pharma companies, but the incentives line up such that you kind of want to be working on things that are chronic or things that you can dose over and over again, rather than things that are actually preventative. And so as a result, vaccines are considered to be relatively unprofitable as an investment for a big pharma company to make. I always think this is an ironic point in the context of what you hear about vaccines, which is that they're sort of a big pharma conspiracy. If anything, it's the opposite. The incentives do not align for big pharma companies to invest in vaccines. They align for them to invest in nearly everything else. And in particular, pathogens that are very relevant in developing countries, in places like India, where I grew up and live now, tuberculosis, hepatitis C, and so on, are irrelevant in places like the US, or not as relevant. And so the likelihood of being able to recoup this investment of $1 billion is extremely low. So as a result of this, we still have these 3 pathogens, among others, but in particular these 3 pathogens, tuberculosis in adults, strep A, and hep C, which cumulatively claim 2 million lives each year across the world. A good chunk of these are in India, a good chunk of them are in other developing countries. Some of them are in rich countries, especially for hep C, but this is just a staggering death toll from a set of pathogens that I think we really ought to have solved by now. And so that's where PopVax comes in. We are funded by an eclectic group of individuals that don't always agree with each other. Vitalik was our first major funder. We're subsequently funded by BARDA, the US government, also by the Gates Foundation. And in the beginning, it was mostly to fund a broadly protective COVID-19 vaccine. So as many of you probably know, the COVID vaccines initially were effective against the wild-type variant, and then subsequently, as mutations stacked up, these vaccines lost their efficacy. And so there were these new variant-adapted vaccines that came out step by step. Unfortunately, these vaccines didn't really come out in a lot of developing countries. India, for example, didn't have any variant-adapted vaccines. And you had to keep going in some cases every 6 months as new variants came out to keep getting these vaccines, which were less and less efficacious. So the question we asked ourselves was, if it is indeed the case that the design of vaccines or the design of these kinds of prophylactics has fundamentally changed with the kind of the advent of generative AI tools, the same kind of diffusion models that you use for image generation can also be used to generate proteins. The same transformers that you use for LLMs can also be used in the context of AlphaFold to predict the structure of proteins. Is there something fundamentally different we can do to make vaccines that are more broadly protective? And we found that there are, in fact, a bunch of relatively low-hanging fruit that you can use to make vaccines that are more efficacious and broader. And I'll explain one of them, which is basically, at the top, what you can see there is the structure of our broadly protective COVID-19 vaccine candidate, which is mounted on a self-assembling virus-like particle. What's in green is the RBD immunogen, which comes from the pathogen. What's in blue is a self-assembling protein that comes from a bacteria. So virus-like particle vaccines of this kind are massively more immunogenic. They can be much more potent than vaccines that don't use this technology. And you can see that here in our data, which is that compared to an existing kind of first-generation COVID vaccine that was approved in the US, we were able to get something like 22 times the neutralizing antibody titer in a pseudovirus assay with the same amount of mRNA. But these vaccines are actually quite hard to produce. So if you want to produce a vaccine like Gardasil, the HPV vaccine, this takes decades to refine the process for. The actual production process for that vaccine actually takes more than 2 years end to end. What we did is we found a way to encode this into mRNA so that you can produce something that's as simple and fast to produce as mRNA LNP, which is the most efficient vaccine production process. But you still get these immunological benefits that you see with virus-like particles. And in addition to this potency, we also saw breadth, which is to say, as you can see on the bottom, we saw that we were able to, with a wild-type-based vaccine, neutralize all these new variants that had made the vaccines ineffective. So there's something about the way we were presenting the immunogen, and we did make some modifications to the RBD as well. But to simplify, there's something in the way that we were presenting this immunogen that resulted in the induction of a more broad set of antibody responses that were relevant against new variants. So we demonstrated some of this early data starting in about 2022. Nobody really wanted to fund this. So VCs didn't really believe that there was the talent in India to actually advance this product. The Gates Foundation initially bet only $100K on us from what I later learned was their publicity budget. So they weren't particularly serious about, in the early days, funding us deeply. And we basically went bankrupt pretty quickly, even though we had generated some very promising data. So this is when we encountered Vitalik, and he made a bet on our vaccine and on the company on the condition that we make this vaccine open source so that other people could build on it in the future if there was another COVID variant or a new pandemic created by a beta coronavirus similar to what we've seen with SARS-1 and SARS-2 and MERS-CoV. Then other people could build on this technology and kind of transparently manufacture this technology. And so we made that agreement with them. We have advanced this vaccine forward with the intention that when we take it to phase 1 clinical trials, we'll release all of the information about the manufacturing and about sort of the design of this vaccine openly, such that others can advance this technology independently of us. So beyond this vaccine program, we have this ambitious million lives mission, which is intended to go after these exact same pathogens I was talking about: TB, strep A, malaria, hepatitis C. So malaria, they're the existing vaccines. We want to make better vaccines for malaria. The existing vaccines aren't as efficacious as they perhaps could be, with the intention of saving over 1 million lives per year if we're able to make effective vaccines against these pathogens. And in hepatitis C, using many similar technologies and using machine learning to design and iterate on these immunogens, we've been able to see a stunning increase in the potency of our vaccine candidate just over the course of the last 1 year. So we see that there is now the possibility, by using machine learning to design these immunogens and then iterating on them based on wet lab data, to be able to make them better and better in a way that hasn't been possible with these pathogens before. The other big advantage for us is we're able to do this much, much cheaper in India. So this is a real comparison between what a US vaccine startup, that's kind of a peer of ours from, say, a decade ago, and in roughly the same time in history for the company, spent on getting one vaccine into phase 1. So it's about $300 million end to end. Whereas we've spent over the last 4 years just about $20 million. We have a whole preclinical pipeline of a bunch of vaccine candidates. And we'll have our first vaccine in clinic now in phase 1 in Australia in 2 months now. So we now want to see if we can do this even faster. Like, can we actually move even quicker than we'd planned to using Prospera, using regulatory frameworks like this? And one potential idea is, could we have a whole bunch of vaccine clinical trials going on at the same time? Could we bring in a bunch of volunteers who are interested in serving as the first few people, so just n equals 5 or n equals to 10, to give early safety and immunogenicity signals for example, the first new HCV vaccine in a very long time, or the first new, kind of new concept of a TB vaccine developed in a very long time. Even if these vaccines wouldn't be effective on the first time round, we have a reasonable expectation that they could be made to be safe using preclinical data that's similar to what we've done for our other program. And this early data would give us the ability to further iterate on these immunogens and iterate on these designs to make them more effective the next time round. And I think this is a critical, piece of philosophy that something like Prospera can enable that other systems can't enable, which is the clinic isn't just the endpoint of your journey, right? The clinic is part of your experimental iteration process. As long as you can make it safe to inject something, you can get information out about the efficacy of that thing, or at least the potential efficacy of that thing, by having different versions of it that you put into clinic at the same time, or iterate over a period of time. And that's something that used to happen under sort of previous conceptions of how regulatory systems for medicines worked, but it's something that is now difficult to do under the current regimes, just because the cost is too high of doing that iteration. We're also working on personalized cancer immunotherapies. This is some early data we have of a cancer antigen from liver cancer, where we're able to generate, with a hit rate of 60%, fully de novo proteins that are able to bind this antigen and turn them into mRNA-encoded, what we call hydaptors, that are able to redirect T cells to kill these particular liver cancer cell lines. And this is something we're now testing in vivo and testing in more appropriate models. But I think what this demonstrates is we can actually take many of the same ideas that we had in vaccines and start to apply those same technologies to immunotherapy, not just for one single antigen, but across a broad swath of antigens that are specific to each individual tumor. So you've probably all heard of these personalized cancer vaccines that people are working on. There's this sort of dog cancer vaccine story that everybody's been circulating for the last 2 weeks. The idea here is not just to do this for vaccines, but to actually do this for more powerful immunotherapies. Something that's almost as powerful as CAR T, but that you can deliver without having to go and re-engineer cells. And this is a powerful idea that we think we'll be able to take to at least some clinical trials over the next 1 or 2 years. And in Prospera, I think this is another thing that could happen that can't particularly easily happen in other parts of the world, which is you could actually charge people for these kinds of personalized therapies right away and use that to fund the development of these therapies. Unfortunately, I think infrastructurally it's not quite there yet. And I think there's some ongoing conversations about, is it possible to have some kind of research hospital or some kind of immunotherapy phase 1 facility within the bounds of Prospera that is able to handle cytokine release syndrome, or able to handle neurotoxicity or other potential off-target effects that could happen with these therapies. I think if that happens, then it might be possible to basically leapfrog what will otherwise be, I think, a decade-long slog to get regulatory systems to recognize these kinds of personalized medicines and sort of pull the future towards where we are now. So I'm very excited about the possibility of doing that here and the possibility of sort of helping enable some of this work to be accelerated. And I'm very happy to collaborate with anybody that has ideas in this space or has thoughts on how this could be done. I think these are all early thoughts for us. We don't quite know how we want to use Prospera, and we don't quite know how the Prospera ecosystem can benefit us. I think at the same time, we don't know how we can best benefit the Prospera ecosystem and how we can best sort of enable more interesting things to happen here. And so we're very excited about finding the right ways to do both of those things. Thank you, everybody.
Super fascinating. Could you talk a bit about the open source aspect?
Yeah, so it's, I think, we, obviously the COVID-19 vaccine we developed is now not as perhaps relevant as when we first developed it for the moment, because we're not seeing this massive surge of COVID-19 anymore. We're not seeing other beta coronaviruses yet emerge. But this is a family of pathogens that has had a long history of coming and attacking the human species, whether with SARS-1, for those of us who were in Asia at that time, obviously SARS-2 globally, the potential of MERS-CoV and these other beta coronaviruses. So I think this is like an ideal candidate for something where the monetary value for us of releasing this open source, it's not going to hurt us that much as a company. In fact, if anything, I think it demonstrates that we can open up our science and be transparent about it in a way that is credible. At the same time, I think it will genuinely help future biosecurity efforts if another beta coronavirus does come to attack us, which it frankly might well, given the history here. And it'll give people a kind of base to build on, especially people that aren't necessarily in rich countries that already have these technologies. But the next time we see one of these global pandemics, if it's a beta coronavirus, African vaccine companies or researchers, or here in Latin America, or in Southeast Asia, where those folks weren't able to develop those vaccines indigenously, might now have a fighting chance to be able to do that. Because we're not just open sourcing the design, we're open sourcing the entire kind of manufacturing schema and quality control schema, which is really where the gap often lies in places that don't have a lot of experience in biologic manufacturing.
Can we see the 300 million versus 20 million slide again?
Oh, sure.
Follow-up question?
I just want to take a picture of it. Can I take a picture of it?
Yeah, go ahead. And this, obviously, to be clear, these are from one specific example. I don't think you can extrapolate this completely across everything that might be possible. But I think it does demonstrate what the gulf is and the cost of development. It is just very expensive in Boston or San Francisco to do even the preclinical development, forget even going to phase 1, 2.
You mentioned the business challenge of vaccine development.
Uh-huh.
How can this be improved? How can it be more incentivized?
I think hopefully we will see people start to recognize vaccines for what they are, which is longevity medicines. If you think about what we've learned about what viruses can do and chronic pathogens can do over the last 5, 10 years, one of the most interesting things we've learned is that Epstein-Barr virus seems to directly cause multiple sclerosis, right? We've also seen, in the last 50 years that we see links between things like HPV and cervical cancer that have been unearthed. I think the likelihood is there are a lot more of these linkages between pathogens and downstream chronic conditions than people expect. And there are potentially business models where one could essentially sort of pay for the reduction in long-term treatment that one might get from these vaccines if that turned out to be true. If it turned out that you could prevent multiple sclerosis using an EBV vaccine. And people have looked at models like this for gene therapy. Maybe that's something that could be applied to vaccines as well. There's also kind of the public goods angle. Obviously, we have been funded in many ways as a public good for our COVID-19 vaccine. I think there should be more funding of this kind for not just vaccines, but for other medicines, which are, they help public health a great deal. They sort of move the ball on longevity and on patient health, but they're not particularly profitable for the companies that are making them. So prize pools and some kind of public goods funding mechanism there, I think, is useful to have. So I think some combination of those two, where you both push on the private sector side, there are lots of benefits that health systems and that individuals are gaining from these vaccines that aren't priced into how much people pay for them relative to other medicines. And at the same time, are there ways to, even if it's not profitable, to continue development of these medicines? I think both of those are particularly good. I will also say, I think funding vaccine developers in developing countries, which ironically is something that hasn't happened for new development as much. People fund vaccine developers in India to adapt an existing vaccine to a low-cost model that can be then distributed in high volume. They haven't typically, funded Indian vaccine companies to develop completely new vaccines. Because of this cost efficiency, even if there's kind of less money to move around, for a company like us, it may be profitable to develop a vaccine that it's not profitable for Merck or Pfizer to. If it's only going to cost me $100 to $200 million to get an Hep C vaccine approved in India, and I think the vaccine is going to make a maximum of a few $100 million a year across the world, that's an investment that maybe I'm willing to make, right? But it's not an investment that Pfizer would make if it's going to cost Pfizer $2 billion to develop a vaccine that's only going to recoup them a few $1 billion over a couple of decades. So I think changing where these are developed, which biotech has really been concentrated in just a few places. It's really just been, to some extent, the UK, Boston NSF for the last 30, 40 years. Now China, to some extent, we're seeing the growth of sort of Chinese biologic developers. But helping sort of move some funding to places where the cost of development is fundamentally lower changes the economics of what's profitable.
Thank you, Soham.
Thank you.